Thank you very much. I certainly can speak for myself, and I think for Brian, how enormously honored we both are to be the recipients of this prize. I want to also thank the Sjöberg family, the foundation, the committee, and the Institute, as well as the Academy, for recognizing the work that we have been involved in. But I must confess that we stand on the shoulders of giants and have used information from scientists that have gone decades before us to make the advances that we made. And we just, in large part, have been at the right place at the right time to translate some of these things into clinical reality. I will tell you briefly about two stories that I think underscore what the possibilities may be, and I think speak to the marriage of clinical research and basic research in a translational milieu that allows rapid development and movement of new information into clinical reality. There are two drugs that our lab has been involved in. I don't pretend for a moment that we were the only ones involved in development. Other colleagues were involved in clinical trials and preclinical medicine, but again, we happened to be in the right place at the right time and performed some fundamental studies that told us that we should just continue to pursue the data, that the data were correct, and that the reality was, irrespective of skepticism, that these alterations could be targeted and they could lead to tailored therapy as opposed to the one-size-fits-all therapy we were using previously. The two drugs are, of course, Trastuzumab and the CDK inhibitors, in this case, palbociclib, but as you all know, there are two other drugs that have been approved in this class as well. We set up a laboratory, and this was based on the work we did with HER2, that really was called the Translational Oncology Research Lab, that using the platform's information that we learned from the HER2 story, we wanted to begin to look at other cancers. And that's how we began to understand what might happen with other kinase inhibitors like the CDK4/6 inhibitor. We also engaged a group of clinical trialists from across the country that were interested in trying new therapies early in their development, as opposed to simply looking at what was coming out of the cooperative group studies and what was coming out of industry through CROs. And this translational platform allowed us to move things rapidly along. We're doing something that I think many places, including and especially places like the Karolinska, are doing, where we use our basic science preclinical models to develop data that will allow us to have information on which we can think about protocol development, moving that to the clinical teams, but totally engage with the basic scientists to tell them what needs to be looked at, how the data informs that we might develop a clinical trial, and then actually develop that protocol and execute on it. But this is an iterative process. It is not a one-way street from the lab to the clinic. While the clinical trials are being executed, material from the patients come back to the clinic to begin to inform us if we're seeing responses, why, and if we're seeing resistance, why. So again, this tends to be a circular and important part of translational research. It is by no means a one-way street. We got involved in the HER2 program in 1986 when I received a cassette of about 6 genes that had recently been cloned by Axel Ulrich and his colleagues at Genentech. One of those genes was a gene called HER2, human epidermal growth factor receptor number 2. That would indicate there must be a number 1. The number 1 was, of course, the EGF receptor. And there are 2 other members of the family, HER3 and HER4. So scientists have been very clever in their nomenclature. And there are 4 members of these receptors that interact one with another and signal from the outside to the inside of the cell in terms of growth potential and differentiation. For this part of the story, we actually started with clinical material and built our models subsequently. And using clinical material, we were early in the idea that we needed to use clinical material that had annotation for which there was clinical follow-up, so that if we found any kind of alteration, we would be able to correlate that back to what the clinical information told us. This next slide is something that I have to explain to new postdocs and graduate students because they have no idea what a southern blot is, because we were using very primitive technology at the time when we started this work. This is the work that first told us that there might be an important alteration in approximately 20% of breast cancer. This represents a series of different patients' breast cancers. Each lane is a separate cancer. Exactly the same amount of DNA has been loaded into each lane, and then we probe the southern blots with a HER2 probe, looking for gross alterations. So again, this is very primitive technology by today's standards, and we would only be able to pick up gross alterations if they were present. And when I mentioned early in my introduction about being fortunate, this indeed was a gross alteration that stood out quite readily. So what we found in tumors that did not have the alteration, as well as normal tissues throughout the body, was a signal that looked very much like this, or this, or this. But as you can see, there are many tumors that have a signal that's much greater in intensity, indicating the presence of a genetic alteration known as gene amplification. So for reasons that we still don't understand today, the Xerox machinery copying the DNA gets stuck on a piece of chromosome 17q and makes multiple passes, indicating you get multiple copies of the gene, anywhere from a 2 to 5-fold increase, a 5 to 20-fold increase, or in some instances, a greater than 20-fold increase. Now, when this gene is amplified, we saw just another manifestation of what cancer scientists already knew, that there is an instability in the cancer genome. That led to the age-old and time-honored question of, so what? What have you shown that is new? But we had clinical annotated material, so we could go back to the clinical material and ask, was there any association? And that is where the excitement started, because we found that in patients who did not have amplification versus those who did, the outcome data in these Kaplan-Meier curves for both disease-free survival and overall survival was very different, and that this was an independent predictive factor, that if the patient's tumor contained this alteration, which is a non-inherited alteration, it occurs sometime during the life of the individual, again for reasons we don't fully understand, but when it's present, and it's present specifically in the tumor tissue, the outcome data are different when the patients are treated with our one-size-fits-all approach. We then wanted to ask if the gene was amplified, was it expressed? Because many times in these amplicons, there are genes that are carried that are part of the amplicon but are not overexpressed. And what we found, again using clinical material, was there was a direct association between amplification and overexpression of the gene product at the RNA protein level that could be measured by northern blotting, western blotting, or by immunohistochemistry, where you see a tumor that does not have the alteration, a tumor that has lower levels of amplification but is amplified, intermediate levels, and very high levels, that the amount of the protein displayed on the tumor cells is much greater, commensurate with the amplification. That then defined this new group of breast cancers called HER2-positive breast cancer, and really set the stage for what we currently use as our 3 therapeutic subtypes. There are many more molecular subtypes of breast cancer, but there are 3 major therapeutic subtypes. Of course, the hormone receptor-positive subtype, which is the greatest number of breast cancers, comprising about 60 to 65%. The HER2 alteration, approximately 20% of those. And then it defined a new group by what they did not have. That is, they did not have amplification of the HER2 gene, and they did not have appreciable levels of hormone receptors, the so-called triple-negative population. Now, it is absolutely true that within each of these large subgroups, there's dramatic heterogeneity and other alterations that undoubtedly play a role in pathogenesis, and we're still working very hard on understanding that. But the reality is there are 3 major therapeutic groups that we're dealing with breast cancer today, based on the observation that there was this new HER2 group and triple-negative group. Because with the clinical data, we know that, again, the outcome for those patients who had the amplification and overexpression of the protein was quite different. This is now median survival for patients from initial diagnosis. And this is a node-positive cohort. Median survival for this study was 6.8 years for patients who were HER2 normal. Of course, some may live much longer, 20, 30 years, others shorter. But this is the median. But for those that had any level of amplification overexpression, the median was less than half, despite receiving exactly the same treatment. And they were, this was an independent predictive factor, independent of nodal status, size of tumor, or grade. This was controversial when we initially published it because people using other technologies didn't find the same level of amplification, but subsequently with more refined technology, the groups of Fenfeer, the Norwegian study group collaborating with the Stanford data set, found essentially the same thing we found. With the Fenfeer data set, the Dutch data set, these patients were tumors that were taken from patients prior to the introduction of adjuvant chemotherapy. So these patients only received surgery and some received radiation. None received endocrine or chemotherapy. And when the molecular analyses were performed on those tumors taken out of the freezer, it was found that the natural history and the outcome data was quite different depending on the subtype. This being the ER positive, so they had among the best prognosis in terms of recurrence or death. This is the luminal B, also hormone receptor positive, but having other alterations in the hormone pathway that make it less hormonally responsive. This is the so-called triple negative that don't have hormone receptors of the HER2 alteration, and these are the HER2 amplified tumors. And this is even after applying therapy to these patients. These two subgroups still remain with a worse outcome, with patients succumbing with a recurrence in general within 4 or 5 years, and an overall survival as seen here after metastatic disease develops in a very short period of time. That led to the obvious question of why. Why was this tracking with such a bad outcome? Is this simply a useful flag of aggressive tumors? Or is it tracking with aggressive tumors because it's playing some role in that pathophysiology? And that led us to do model studies. And that's how we built the platforms that I'll finish up my lecture with in terms of HER2 and then subsequently CDK4/6. The breast cancer panel we put together was some 51 cell lines, and I'll talk about that in a moment. And the idea was to try and use models because, well, the tumors told us the presence of the alteration, and the clinical data told us the association of the alteration with outcome. There's no way to manipulate those tumors in real time, but you can manipulate models, cell lines in the laboratory, or human tumors growing in experimental models as xenografts. So we wanted to build a panel that initially started with the HER2 story in breast, but is now extended to some 620 cell lines across some 16 histologies. And again, the numbers, we're not trying to get just huge numbers of cell lines for the pleasure of collecting cell lines, but we're trying to capture as best we can the molecular heterogeneity of the disease that you see in the clinic. The cell lines in this panel are fully characterized for their growth in 2D, for their growth in soft agar, and for their growth in vivo, both subcutaneously and where appropriate orthotopically. We have, as I say, the xenografts from each of those. Not every one of the cell lines will grow xenografts, but we know which from each of the histologies will, so we have a sampling. We also have PDXs from many of them. And finally, we have a tumor tissue bank. And tumor tissue bank should be filled in for all of these, but that is clinical samples. Not necessarily that the clinical samples generated the cell lines, but if we see something in the cell lines, we would then want to ask, do we see the same thing in actual tumor tissue, so that we're not chasing an artifact of laboratory propagation of cells in culture. And we also have frozen and paraffin-embedded microarrays. But finally, what we have in this relational database is the response of the cell lines and the xenografts to the approved therapies in those particular histologies. So all of that's in a relational database that then we can explore. So the cell lines have all been biologically characterized. They are also molecularly characterized in the sense that each cell line in the panel of all 620 have had copy number analysis using CGH arrays across the entire genome for copy number loss and gain in regions of the chromosome. We've also done exome sequencing for mutational analysis, adjuvant microarray analysis, and using the RPPA platform, protein analysis on each of the cell lines, and all of this, again, is entered into the relational database. Once we have that information, then we have, as a platform, what we feel is a good tool to begin to test various inhibitors, like we did with Trastuzumab or Herceptin, or subsequently with the CDK inhibitors. And here are the number of different inhibitors we've run through the panel so far, some 285 against these targets. Some we have only a one-off experiment in terms of a drug against a particular target, but others we have, for example, the EGF receptor tyrosine kinases, multiple shots on goal, as well as like the PI3 kinase pathway. But again, all this is in a relational database. Now, this information could be exciting only if you see differential response. If you see something like this, this is now an IC50 curve. This is a typo. This should be micromolar. And if nothing is responding, they're all bunched together. Each circle represents a separate cell line from a separate tumor, and then breaking it out by histology across the panel. If nothing is working, this is what you see. If the drug is incredibly toxic, this is what you see, where all the cells respond. Where the excitement starts is when you see a picture like this, where some cells are very resistant, where other cells are exquisitely sensitive. Now all that information we put into the database becomes useful. We can ask what is tracking with response and what is tracking with resistance. And this is a RAS-RAF/MEK inhibitor showing that RAS-altered tumors in the cell lines frequently have response to that inhibitor. Well, we did the same thing with the breast cancer group. In terms of the platform, this is what we in fact started with when we found the HER2 alteration. So we had a number of cell lines that were HER2 normal, ER positive, a number that were ER positive and HER2 amplified, ER negative and HER2 amplified, all in the luminal group, some 25, and 28 in the non-luminal group, the normal-like tumors, the basal progenitors, and the mesenchymals, some of which were HER2 amplified as well. And we wanted to ask, was this tracking with a bad outcome because it was playing a role in pathogenesis? And one of the things we did initially was to take this group that did not have the alteration and introduce the alteration. So we take HER2 normal breast cancer cells, clone the gene from a tumor that had the alteration, sequence it to make sure it was just amplification and no mutations in the gene, and then we transfected it into cells that were HER2 normal and expressed normal levels, converting single copy normal expressors into multiple copy high expressors. And we did it for multiple cell lines in both breast and ovarian cancer because we see this alteration in a subgroup of ovarian cancers and gastric cancers. This simply shows we can do it. The parental cells are shown here, the daughter cells here, the expression of the receptor here. The amount of the receptor expressed in these normal cells is about 25 to 50,000 receptors per cell, which sounds like a lot. However, when the alteration occurs in patients, or when we do this in a lab, that number goes up to greater than 1 million. So there's a 2-log increase in the amount of receptor. Otherwise, the cells are genetically identical, except one has the alteration and one does not. That allows us to assess the impact on biologic activity. And what we found, these are the HER2 normal cells. This is an MCF7 parental cell. These are the exact same cells that have the alteration. This is growth in 2D and plastic over days, a number of cells in a culture, and you can see the proliferative potential of the HER2 positives is dramatically increased over their parental counterpart. If we look at 3D and anchorage-independent growth, the same is true. There are small punctate colonies forming here, but in the HER2 positives, it's much more numerous and much larger. And if we take these and put them into experimental mice, we see this. These are nude mouse xenografts. Both our litamate controls are injected with exactly the same number of tumors cells at the same time. And within 2 weeks, this is the HER2 normal tumor growing, and this is the HER2 positive tumor growing. It's much larger. If we take these and put them orthotopically in the cleared mammary fat pad, allow a tumor to form, and then remove it surgically, mimicking what we're doing in the clinic, watch the animal after we remove the primary tumor, the metastatic potential of the HER2 positives increases by 225%. That told us the reason this was tracking with a bad outcome was it was playing a fundamental role in causing that bad outcome. The HER2 alteration significantly increases growth by any number of measures, metastatic potential, and alters the response to hormonal manipulation. That led to a therapeutic model and the ultimate validation, which is if we were correct, inhibiting this alteration should reverse some of those phenotypic phenomena. And this is some of the initial data we generated with an antibody directed against the extracellular domain of the receptor, just adding small amounts of this antibody, 4D5, to the culture resulted in a precipitous drop in the proliferation rate and a G1S arrest of those tumors. And with prolonged exposure, those cells will undergo senescence and die. This is unique to the HER2 positive cells. The reason of having the panels, we could test it in the HER2 normals and ask, would the drug have any impact there? And it does not. That told us there may be a therapeutic window in the clinic. This is the nude mice that are growing the tumors. As you saw, those tumors grow quite rapidly in time to large tumors in the animal. But if we administer the drug, it can be completely suppressed. And with these data, we convinced some of the colleagues at Genentech that the antibody should be humanized, and then took this data to the FDA and asked for permission to begin phase 1 clinical trials in patients. And that was where we started the clinical translation, and the hope was that we would be able to use this biologically, what we felt was biologically relevant molecular information, to lead to a more effective and hopefully less toxic therapy. The phase 1 program used the mouse monoclonal antibody. The initial patients treated this were treated with doses less than we gave a mouse. 120 micrograms was given to a 70-kilogram woman when we were giving a 30-gram mouse 150 micrograms twice a week. Why would we be conservative like this? Well, because as I mentioned earlier in the lecture, this receptor is expressed normally in multiple tissues throughout the body, and the concern was we would run into toxicity. In fact, we didn't as we dose escalated. I just want to pay tribute to the women who went into these clinical trials because we told them that went into the earliest phases that they could not receive additional doses because we had to observe them for prolonged periods of time. And that we thought this might even be homeopathic in the informed consent process, but every one of the patients approached said, we understand. And proceeded with moving, excuse me, and when thinking about those patients, it's quite emotional. We understand that we'll go ahead and allow you to dose escalate to the further patients. And we showed safety, and we showed evidence of efficacy with the mouse monoclonal antibody. Now, the only thing we did see was that this mouse monoclonal antibody generated an immune response. So the mouse antibody was recognized as foreign, and we were able to convince at that point Len Presta and the group at Genentech to humanize this to the human form of 45, which ultimately became Trastuzumab, but a drug you know as Herceptin. We entered that into phase 1 trials, both as monotherapy and in combination with chemotherapy. And we found that we were getting evidence of signals, and it was quite safe. We saw absolutely no toxicity. That led to the design of the registrational trial of 460 plus patients who had metastatic disease that were HER2 positive, and we now tailored it to that positive population. Primary endpoint was time to disease progression. Secondary endpoints are shown here, including survival. What we found at a very early response time in the trial, in months, is the curves separated significantly when we are adding this to the standard treatment. And what we found even further is we improved the response rates by 50%, the response duration by 60%, and the time to progression, which was primary endpoint, by more than 2/3. And this over chemotherapy. This met the primary endpoint, but even more importantly, what we saw at the 1-year mark was this. Patients who received the antibody were 78% of the patients were alive versus 67% who received chemotherapy alone, despite the fact that we had biased the trial against seeing this. Why would we do something like that? Well, for ethical reasons. The patients that were randomized to the control arm were told in the informed consent, if your tumor progresses and if you want the experimental drug, you can receive it. More than 2/3 of the patients in the control arm elected the crossover. They crossed over and 1/3 of those responded. We would have seen a greater difference in survival had we not allowed that. So despite that, we still saw what was a significant difference. And that led to the current state of affairs for HER2 positive breast cancer. We now know for metastatic breast cancer, those patients have gone from having the worst outcome to now having among the best outcome in the post-Herceptin era, even better in the early stages, certainly, than HER2 normal patients, and certainly better long-term in the post-Herceptin era than when we had no Trastuzumab. However, in early breast cancer, it becomes even more dramatic. In early breast cancer, and this is data from Carlos Barrios, who is a collaborator in Brazil, who was looking at natural history data in breast cancer in patients who are treated with just surgery alone. And you can see when you start the observation period, all are alive, but after about 13 to 15 years, only 26% are alive without recurrence or death. When Gianni Bonadonna did his pioneering work of adjuvant chemotherapy, that improved by this percent. When other things like anthracyclines were added, that improved things yet a bit further, but added some additional toxicity. And it wasn't until the introduction of the taxanes in chemotherapy that there was a significant improvement in outcome. So the reason the curves don't go out this far is because these drugs were introduced after the combination CMF regimens and anthracycline regimens. Dose-dense alterations can modify this a bit, but again, these are small differences. But what Herceptin has added in the early breast cancer treatment is patients, rather than having a recurrence and death in 5 years, we are seeing this. So the drug was approved in metastatic disease in 1998. It was approved in early disease in 2005. And now this is what we're seeing in terms of the natural history. So it has indeed been reversed using this targeted therapy. I'll finish up on the CDK4/6 program with just a couple of slides there. Again, using the panel, we looked at a CDK4/6 inhibitor that was developed by Park Davis. This drug was sitting on the shelf at Pfizer that acquired the compound. It had been tried in mantle cell leukemia and some childhood sarcomas without significant success, so it was shelved. And that prompted this editorial once we showed the clinical data in breast. This was an editorial in Science talking about the cancer drug that almost wasn't. And again, this is a cell cycle inhibitor that inhibits CDK4/6. Now, as you all know, CDK inhibitors have been looked at for a while, but the earliest shots on goal, the earliest inhibitors were pan-CDK inhibitors, and they resulted in significant toxicity and really never moved very far out of phase 1 testing. The drug we took initially was palbociclib. This drug shown here, it is among the first in class. It is specific for CDK4 and 6. It does not hit the other CDKs. It works as inhibiting the kinase activity of this enzyme, And when it couples with cyclin D, which it does normally, and carries out hyperphosphorylation of RB, it releases cells from the restriction point and allows them to progress through the cell cycle. What happens is if you inhibit this enzyme, you do not get hyperphosphorylation and the cells are stopped here. And with prolonged exposure, in theory, you might undergo senescence. We were interested in this drug. This simply is the mechanism of the drug. There's a natural inhibitor in the body of P16, which is an inhibitor of the CDK4/6 cyclin D combo. And then, as I said, hyperphosphorylation of RB is what allows the cell to progress. We ran this in a panel, and what we saw was something very interesting. Again, it didn't look like saline, and it didn't look like bleach, but it gave us the spread. And the spread was really significant, and I know it's hard to see the light pink on white, but it was the breast cancer panel within the group that was really showing the signal. That told us that this may be where the action was at, and we went ahead and looked at, this is now the bar graph of that same IC50. The bigger the bar, the more the drug, the smaller the bar, the less amount of drug to get an IC50. And the breast panel broke itself out into 3 groups: highly resistant, intermediately sensitive, and exquisitely sensitive. When we unblinded the data, we found that the cell lines that were exquisitely sensitive were in fact the luminal ER positive. Now, my preconceived notion going into this experiment was that triple negatives would be responsive, and I was completely wrong. In fact, they were the ones that were relatively resistant. But we did this in an unbiased way and let the data tell us where the story may be. Now, we saw this and wondered why were we seeing this, and that led to going back to the other data in that relational database for the cell lines. And when we analyzed that, it became apparent why we saw this. This is the only microarray data I'll show you, I promise, and it is, now the bar graphs are turned on their head. Still, the bigger the bar, the more resistant. The smaller the bar, the more sensitive. And second from the bottom is the relative expression of retinoblastoma in these cells. And it is the luminal ER positive group that is expressing the highest levels in the panel. And in fact, the triple negatives are expressing among the lowest levels. And if you look at P16, it's almost the exact reverse. The expression of P16 shown here versus here. That explained why we were seeing what we were seeing. And that led to the idea that we might be able to target this in ER-positive breast cancer. We translated that into the clinic now to try and benefit this group of breast cancer patients that have luminal A or luminal B disease. And what we have been doing for them for the last basically 45 years is attacking the estrogen receptor pathway. The question was, could we attack a pathway outside based on these data? So we asked for permission to do an investigator-initiated phase 1 trial. Pfizer gave us the drug. We entered 12 patients into this phase 1B trial to see if we could safely combine them. And in fact, we saw that we could give it safely, but more importantly, we began to see responses. And we saw, of these, 2 of them went on to complete responses as we watched them further. And at that point, Pfizer saw the data and said, this is no longer investigator-initiated. We are pulling it back into the company, but you can run the clinical trial, which we did in the TRIO network. And the clinical trial for the phase 2 design was broken into 2 groups. 60 patients were supposed to be enrolled for ER positivity alone, HER2 normal. We tried to enhance the selection even better with cyclin amplification or P16 loss, and another 160 were supposed to be enrolled there. When we finished the enrollment of the first 60, we overshot by 6 patients, and started the enrollment of the next cohort, we saw these data. Patients were coming off study because their disease was progressing in an almost 2-to-1 ratio if they were receiving the best available standard therapy versus if they got the CDK inhibitor. That led to looking at the Kaplan-Meier curve, which showed us this. There was a dramatic difference in progression-free survival. Now, when I first saw this data, of course, I was happy, but I was a bit skeptical because this is not a typical response for an aromatase inhibitor. in ER-positive breast cancer, 5.7 months versus 18 months. But these are randomized data, so we were convinced that they were likely to be correct. It's just that we're small numbers. But remember, in this design, we had designed the second part. Pfizer stopped the whole trial when we saw the signal and said we are ready for phase 3. But by then we had enrolled 99 patients treated exactly the same way. So we had a total of 165 patients that we could do a combined analysis on, and this is what we saw. The impact, in fact, was correct. There was a doubling of the progression-free survival from 10 months to 20 months. This is the largest impact seen in metastatic hormone receptor-positive breast cancer in the last 4.5 decades. So these were very gratifying data. There is a safety signal that is an on-target effect in the bone marrow, but the cells are not killed in the bone marrow. They're simply suppressed. The patients are treated 3 weeks with the drug and 1 week off. In that 1 week off, they recover. And the FDA looked at these data and said, we will grant breakthrough designation, and said we'll consider granting conditional approval if you enroll a trial that's 4 times larger. You don't have to wait to the readout of that trial, but it has to be completely enrolled before we give conditional approval. So the TRIO group, in fact, did that. This is the study design of Paloma 2. It's identical to what you saw in Paloma 1. The statistical considerations are shown here, and these are what the data show. Essentially, it was the same, a 10-month improvement of progression-free survival. Comparing the 2 trials shows that there is almost total overlap in the improvement we saw. These were extremely gratifying data because 2 separate independent clinical trials showed us exactly the same data. There have been 2 other drugs in the class, ribociclib and abemaciclib, that also now show the same thing. All patients across all subgroup of hormone receptor-positive disease benefited, whether they were older or younger, whether they had visceral or non-visceral disease, despite where they came from, whether they had a long disease-free interval or a short one, what their performance status was, and whether they had prior chemotherapy or not, or prior hormonal therapy or not. So these, again, were the data that allowed full approval by the FDA and ultimately the EMEA. So this drug has now recently been approved. Again, the on-target toxicity is what is seen here. We saw no evidence of febrile neutropenia, just neutropenia, and no sepsis. So the story evolved this way. We got the drug in, the drug was developed in the late 90s. We got it in 2008 as part of this work with Pfizer and published on the preclinical work in 2009. Started the phase 1 trial with 12 patients in 2010 and got the signal within short order that launched the phase 2 trial design. When the FDA saw that initial split in the curves, they gave breakthrough therapy status and agreed to give conditional approval. We then enrolled the 4-time larger clinical trial, and the phase 2 confirmed, the combined analysis confirmed that, and ultimately the phase 3 trial confirmed exactly what we saw, and the drug was approved. And since then, 2 drugs again, ribo and abemaciclib, have been approved in this space. I will definitely finish. I'm sure I'm over time on this. When we compare where we're at previously with modern day chemotherapy, cytotoxics, in terms of response rate, in these trials that are shown here and reported in the national meetings with either paclitaxel, docetaxel, or combination chemotherapy or capecitabine, these are the overall response rates from some of the more recent trials. This is what we're seeing. Oh, I went the wrong way. This is what we're seeing with the addition of these CDK4/6 inhibitors. We are now improving overall response rate, but even more importantly, if you look at progression-free survival, this is what we see with the chemotherapies. This is what we've been able to achieve in terms of improvement of progression-free survival with hormonal therapy, and this is what we're now seeing with the CDK inhibitors. So we're very excited about these data. I think more information will be available on overall survival at this year's ASCO. This is the final slide. We now have treated some 300,000 women globally with the CDK inhibitors. Herceptin has been around a lot longer, so almost 3 million women have received this drug. Not all respond either in this group to Herceptin or this group to the CDK inhibitor. So there's still much work to do to find out what defines that heterogeneity. And there is absolute work to do on what the president of the Institute said about the cost of these drugs in terms of making effective agents available for patients who need them, who otherwise aren't getting them because of cost. These are the people in my lab over the last 30 to 35 years that are the reason I'm standing here. And these are the people that I've collaborated with, and I want to particularly acknowledge the Revlon UCLA Women's Cancer Program that was launched by Lily Tartikoff, who is also here to celebrate with us in terms of what the program has produced, and Ron Perlman, who produced that money. Thank you very much for your attention.